So thank you so much for having us here. So myself and Merete will present our chip-based microscope system here today. So I guess we can just get on with it. Or maybe first we should start by bragging a little bit about the Wiley Analytical Science Award, which we were just received the first place in. So a little bit of bragging there, and now I will get on with my talk. So a little bit background about us. So we are from Tromsø in northern Norway. So it started this project around 10 years ago in the University of Tromsø, and then we in 2019 we realized that there was quite a bit of interest in this technology that I will present now. So we decided to open a company, NanoImaging. And now in 2023, we have our first product ready. It's a microscope system, ACP0. And what we do is chip-based fluorescence imaging, both super resolution and diffraction-limited, where we can increase the field of view of the microscope images. So first, since we are talking about super resolution microscopy, I always show this image because it's my favorite super resolution image. So it's a liver sample. It's just to showcase the importance that super resolution microscopy can have to your research. So on the left side, it's the liver sample. These are these endothelial cells, which is lining the blood vessels in your liver. And this is a membrane stain. And in diffraction-limited imaging, you don't see much, right? It's just nothing there. It's on the left side. But with super resolution microscopy, you can visualize this fenestration, these holes in the cells, which are really there to help filter the blood. So that's just to underline the importance of super resolution microscopy. So it comes with a cost, super resolution microscopy, and one of the costs is that you can get this reduction in the field of view, depending a little bit on the technique you use to get super resolution. The field of view can be reduced a little bit. So in this case, it is a single molecule localization microscopy, and typically you need quite a high intensity to get single molecule blinking, and that finally limits the field of view. So here it's an example, 50 microns by 50 microns large field of view, but the content information in the image has a very high resolution. So how can we tackle this problem of increasing the field of view in super resolution microscopy? Well, so what we figured out was that, okay, since you need excitation light to excite your fluorescence molecules, and this is typically done via the microscope objective lens. And then you use typically the same microscope objective lens to collect the fluorescence emission. And what that finally means is that you will limit your field of view. And this is because the objective lens needs to have a high numerical aperture typically. And then you are using the standard glass slide, cover glass, to put your sample, to hold your sample. This increased resolution but limited field of view might lead to you missing features because maybe something happened outside your field of view that was really what you were wanting to see. So microscope technology has evolved quite a lot since its invention. So, but one thing has sort of remained the same, and that is the glass slide. That has remained like that for quite some long time. So here I found online this picture of a microscope glass slide from 1850. It's still a microscope glass slide. It has some nice wrapping paper. They had an eye for detail back in the days. So this is quite nice, but it's still glass. So what we set out to do was to do something with the glass slide. So what we have done is that we are removing the excitation light path out of the microscope. We are still collecting the fluorescence emission with the microscope, so we need the objective lens. But we are illuminating the specimen via this photonic chip. So we are removing the glass slide, we are replacing it with a photonic chip, and this photonic chip serves both to hold the sample, so you are mounting your sample directly on top of photonic chip, And then you can illuminate the specimen via these integrated waveguides, which is on the top surface of the photonic chip. And if you do this, you can have a very large field of view illuminated with a very flat illumination profile. So you can generate uniform illumination. So this is turf illumination. You can generate turf illumination over very, very large fields of view. To see how this works, let's take one of these photonic chips and then do an intersection, cross-section of it, and see how this works. So we have a silicon chip with a few layers on top of it. First is an oxide layer, and then you have what we call a waveguide layer, for instance, silicon nitride in this case. And if the refractive indexes are sort of correct for this, you will get a waveguide in the top surface here. So in the waveguide, you can introduce laser light, and it will be coupled on to the waveguide, and it will be propagated along the waveguide. And wherever there is a waveguide, there's also going to be a little bit of light leaking out of the waveguide. It's called evanescent field, and that's the fundamental basis of turf microscopy. This is normally generated with an objective lens, but you can also generate it this way. And then you can mount your specimen directly on top of this waveguide surface here. And then if they are fluorescently labeled, they will get excited with this evanescent field and emit the fluorescence. And then you can just image this with your microscope from the top. So we have a complete system that takes care of all this, the illumination, the imaging, everything, the software, everything. And this might look like something like this from a little bit of a distance. So you see the stripe of light that is generated, that is the waveguide that is holding a laser illumination inside, and the specimen would then just rest on top of this. So there are some benefits doing it, the illumination, the excitation light this way. Firstly, we have very high refractive index contrast. We are using materials of a high refractive index, and you get a very high index contrast. And then this will lead to a very intense evanescent field. I should also say that the waveguide layer itself is very thin. We use 150 nanometer thin waveguide layers, so you are really compressing the light inside a very shallow waveguide layer with a high refractive index contrast, and this allows the evanescent field to have quite a good intensity. We can make the width, we can scale the width of these waveguides more or less as we wish. So these are made by lithography. So we can make very wide ones to illuminate very large fields of view. With our multimode technology, we can choose which type of illumination we are giving to the specimen. We can have a uniform illumination. We can have a more chaotic stripy illumination. I will explain you what this means. Or there is also a possibility of getting a controlled pattern illumination by using some single mode technology. This will lead to different techniques for imaging. You can have diffraction-limited imaging using uniform illumination, single molecule localization microscopy with uniform illumination. You can do these fluctuation microscopy techniques by using multimode patterns for illumination. And we even have shown that it's possible to do structured illumination microscopy using photonic chips. Structured illumination microscopy is not part of our current product offering, but might be as we move along. Furthermore, you get a very easy multi-channel alignment because you can put different wavelengths into the chip without any effort. I will show you. So what is the real benefit here? Now we will revisit this small field of view with the high resolution I showed you to begin with. It was 50 microns by 50 microns. But since we now have separated the excitation light paths and the detection light path, we are able to increase this with 100 times. So now we have super resolution microscopy. This is single molecule localization microscopy technique over a 100 times larger field of view. and you are maintaining the super resolution here. So it's quite neat. So now you suddenly have the information of hundreds of cells, right? Instead of just a few ones in the same time it takes to do the small field of view. So here is a little bit revisiting some old results back from 2017 that this is really a thing. It works. We have repeatedly been doing this. So this is from a 2017 paper. You can see that the resolution here is around 40 to 50 nanometer. So this is particular result is taken with a 60x objective lens. So the field of view is not as huge as the last image, which was taken with a 25x objective lens, but it proves the point. Multi-channel, as I say, is very easy in our system because you can couple the laser lights directly onto these waveguides from the side, and you don't really need to do any realignment in between each channel. And this I'm showing here, you can see 3 lines. It's green, blue, and red, and at some point it even goes white. So that's all 3 lines together. You don't need to do super resolution if you don't want to. There are many nice applications for diffraction-limited imaging. So here we are showing turf microscopy using a 10x objective lens to gather the fluorescence emission. And you can really see here that it is only the waveguide that is illuminated. And this you see if you look at the yellow here and the magenta, it's membrane stain and actin, I think. And you can see that it suddenly stops. And then you can see the cyan, which is an overlay of epifluorescence and the nucleus stain. So you really see that it is only selectively exciting the structures on top of the waveguide. For those of you who are interested in optics, which I hope is most of you, you would probably realize that maybe this you get some kind of multi-mode things going on here. So at least you know from fiber optics, if you have these different color fibers, you have the ones only holding the single mode, which give you like a Gaussian output, and you have the multimode fiber, the orange ones, which give you speckle output. It is the same with chip-based waveguides. If you have very large cross-section, it will support many modes, and you get a very uneven illumination from this. And we call it multimode interference when you have a lot of these modes starting to talk to each other and create a not so even illumination. And then if you look through your microscope and you have stained the surface of the waveguide with a fluorescent marker to just see how the illumination actually looks like, it might look like this. You see it's not very uniform at all. But you can shift this around, and then these modes, this multimode interference patterns start shifting together, and then you can use this to generate uniform illumination, or you can use it to generate super resolution microscopy. To generate super uniform illumination, we just scramble away these modes very rapidly, so each frame of the output of the camera look uniform like this, compared to if we do not do our multimode scrambling on the left side. You can use this for this fluctuation-based super resolution microscopy. I'm sure you have known all this SOFI method, SOFI or SR surf or musical, or there are many different algorithms. But what they all would like to have as an input is a fluctuation intensity of the fluorescence. So we can generate data like this with static fluorophores. We don't need the fluorophores to shift to fluctuate. We can instead let the excitation light fluctuate by harnessing multimode interference in these waveguides. So the data that you take, a few hundred frames, might look like this, and then you use one of these algorithms. There are many plugins for Fiji. We have our own software also, which has implemented a few of the key ones, which we think are the best. And then you can reconstruct this image into an image with increased resolution, but mostly also it has a very increased contrast.
Hey Markus, kannst du mir doch noch mal kurz zeigen, wie man...
It's both a contrast enhancement and a super resolution method. And you can see the features, the resolution of this image is governed by the properties of the waveguide. This is stated here as NF, which is the effective refractive index of the wave. And that will conclude my part. And now Merete, I think, will show you what this can be used for, applications and very cool images taken. So thank you.
I hope everyone can hear me. I'm just going to share my screen. Can everyone see the screen? Yeah.
Yep, that's coming up and we can hear you.
Perfect. So my name is Merete and I'm an application specialist at Chip NanoImaging. And biology is, it's not, so biology is a bit more varied than what a few cells can show. And I wanted to just exemplify the need for a large field of view using exocytosis as an example. But before I can show the time lapse, I wanted to explain what you can expect. I think this is a laser pointer here. So this is an exocytic event, and what we'll be looking at is granule secretion. And that means that we won't be able to see the vesicle itself, so we can't define this kiss and run or compound fusion events, but we can see how the cargo is being released. So you'll see that it enters the evanescent field and then lingers a little bit before it merges with the plasma membrane and then disperses the fluorescence signal. And this is what that looks like over time if you look at it slice by slice. And the intensity profile is very iconic for this type of event. It looks, why won't it scroll? Okay. It looks like this with a sharp increase and then a slower decrease in intensity. And this is what that time lapse looks like. I had to crop it down a little bit, so this isn't the full field of view, but if you look at it, you'll see that these firework events are occurring a little bit everywhere with varying intensity. And if you look at it for as long as I've looked at it, you'll notice that there are two main hotspots where these exocytic events occur, which you might have missed if you had been using a traditional turf field. So by extending the field of view, we increase the likelihood of event detection. You get more statistical power, and you can also then detect this variation. Now, we achieve this. Oh, it's annoying that I can't. We achieve this by multi-mode waveguide technology, and I won't go too much into it. I think Øystein described it quite nicely. But we then have uniform illumination by using these multi-mode waveguides, and the evanescent field is very intense, penetrating the cells roughly 100 nanometers. And then we provide a complete imaging system with customizable hardware, as well as image acquisition and post-processing software and some consumables. Now, the multi-mode waveguide chips look like this. So we have different sizes for different application needs, and you can seed your cells directly onto the chip surface using these leak-proof, easy to peel away incubation chambers. And if you look at the chip here, you'll see these white lines running in parallel, and that's between these white lines, that's where your waveguide is. So we have different sizes of these waveguides, and this waveguide in the bottom here is 3.3 millimeters wide. So you'll have a uniform illumination for this entire width and length of the waveguide. Now, in addition to cells, you can also image tissue samples, and here you have an example of what that looks like. This is a kidney biopsy where it was a collaboration between the University of Tromsø and the neighboring hospital, where they wanted to visualize this 100 nanometer gap, which is then critical to identify the disease, minimal change disease. And by implementing super resolution, known as, for example, DStorm, they were able to then visualize this gap between podocytes and the epithelial layer, which then helped to inform the clinicians. So a very promising study. Now we can easily switch between epifluorescence and turf mode. So epifluorescence shows you the internal context, and here you see actin in orange and purple showing the internal membrane compartments. Whereas if you switch to turf, you see a very different context. So here the actin is showing you stress fibers, you have focal adhesion points, as well as how the actin is mobilized along the perimeter of the cells. And you can see how the membrane, instead of the internal membrane compartments, you see the plasma membrane and how it's formed these ruffles and slid underneath neighboring cells, for example. Now, because we have both epi and turf modalities, we implemented a deconvolution into our software package, which is a contrast enhancement technique. So you can you can perform two-dimensional deconvolution on both turf and epi, and if you acquire an epifluorescent Z-stack, you can do a projection of your three-dimensional deconvolved stack. And that looks like this, where actin is in blue and the nucleus is in orange. Now, because we use multi-mode waveguides, you can illuminate either with a uniform illumination or this patterned illumination, which is then a multi-mode interference pattern, and it looks a little bit like this. So we have a controller unit that then allows you to collect the different patterns that we can achieve. And then you can use this to reconstruct a super resolved image with improved contrast. And there are, of course, several algorithms available, and we've implemented a few of these. So this is nice because then you can illuminate in a controlled and reproducible manner instead of relying on your biology to fluctuate, because not every fluorophore will have this property. And maybe your cells just didn't want to get out of bed that day. So this is here you can see actin in red, both for the fluorescence fluctuation reconstructed image and the turf image. Now, because we have multi-mode waveguides with tight light confinement and intense evanescent field, it makes it ideal for single molecule localization microscopy, which is also referred to as this blinking microscopy, where you need a lot of power in order to force the fluorophore between this on and off state, in order to isolate it in both time and space. And then you acquire several images, several thousand images, and then you perform a reconstruction where you localize each, the center of each blink, essentially. Now, because we have such a large field of view, we end up with a lot of data. So this is why we implemented a GPU accelerated reconstruction software known as Arctic Storm. And it looks a little bit like this, where you have initial parameters where you can filter out or filter localizations, as well as some post-processing settings where you can filter out uncertain localizations, and you can play around with your reconstructed image. This is what the raw data will look like. And here you have the full reconstructed image. And I'm just going to zoom in a little bit. And then you can see these dots, which are membrane receptors, which then the diameter of which you can kind of isolate or define as monomers, trimers, or dimers. And then you can estimate how the drug has affected the membranes in terms of activity and internalization. So I hope that this has piqued your interest in ultra-large field of view, super resolution, and diffraction-limited imaging. And if you're interested in booking the system, we will be at EMBL early next year. Thank you.